Hey everyone, welcome back to my channel. In today's class, we are going to deal with part 9 of diversity of living organism, that is kingdom Animalia. Let's begin our class with the characteristics of kingdom Animalia. So what are the features present in organisms which are present in the kingdom Animalia? They are eukaryotes or prokaryotes? They are eukaryotes. What are eukaryotes? Eukaryotes are the organisms which have distinct nuclei. When I say distinct nuclei, it means their nucleus is covered with a nuclear membrane. Are they unicellular, multicellular? They are multicellular. What is the mode of nutrition in them? That they have holozoic mode of nutrition. What is the meaning of holozoic mode of nutrition? It is nothing but they are going to ingest food and digestion occur inside their body. Right? Are they heterotrophs or autotrophs? They are heterotrophs. What is the meaning of heterotroph? They depend on others for food. Right? We have already studied this. Now, they have specific body plan. Cell wall is present or absent in animal cell. It is absent in animal cell. It is a feature of plant cell. Right? Photosynthetic pigment, again the feature of plant cell. Now, biology is all of exception. Tell me an animal which possesses photosynthetic pigment. Tissue formation you will see in the organism. Almost in all the organism, tissue formation will be present except for porifera. Phylum porifera, that means sponges. You will see aggregation of the cells, but it is not going to form tissue. Right? Now, mostly animals are motile and they are going to reserve food in the form of glycogen. What is glycogen? Glycogen is nothing but your carbohydrate, that is polysaccharide. Right? So, glucose is going to give us the energy, but what about the extra glucose? Extra glucose are going to form bond with each other through glycosidic bond will be present. Extra glucose are going to link with each other through bond glycosidic, right, glycosidic bond, and these glucose are going to form glycogen. So many glucose are going to combine together with the bond, that is glycosidic bond, and it is going to form glycogen. Mostly animals are motile in nature and some are sedentary or sessile. It means they are going to stay only at particular place. For example, your sponges, I'll show you the picture, and you can see that sponges will not move from its place. Now, muscle cell and nerve cell are the feature of animal kingdom. Have we seen muscle cell and nerve cell in plant? No, we have not seen, right? Reproduction is mostly going to be the sexual reproduction, but can I say that asexual reproduction is absent? No, asexual reproduction is also present, but mostly you see sexual reproduction. It includes simple sedentary, that means sedentary who is going to stay stationary. You can write it over here. Sessile or sedentary means stationary. They are not going to move. Immotile, they are not going to move from one place to another place. So from sponges, it includes from sponges to vertebrate mammalia. All the animals comes under kingdom Animalia, from sponges to vertebrate mammalia. Now, this is the classification table given in your textbook, that is kingdom Animalia, which is also called as metazoa. What are metazoans? Metazoans are nothing but your multicellular organism. Multicellular organisms. Now, these kingdom Animalia is also called as metazoa, and metazoa is nothing but multicellular organisms. Metazoa is further divided into two subkingdoms. What are the two subkingdoms? One is parazoa and second one is eumetazoa. When I talk about the parazoa, para means primitive or old. Primitive or old. What is eu? Eu means true. What is the meaning of zoa? Zoa means animal. So, by this you must be knowing what is parazoa. Parazoa are the primitive animals. When I say primitive animals, that means you won't see tissue, no tissue formation you will see in parazoa. But what about eumetazoa? They are true metazoa. That means you will be able to see formation of tissue in eumetazoa. Okay, they are true multicellular. Eumetazoa is further divided into two. That means two breeds are present over here. That is radiata, which is also called as diploblastica. Okay, either that means the organisms who possess radiata will also possess diploblastica, right? Or bilateral or triploblastica. But before moving ahead with this flow chart, you are supposed to understand what is the meaning of radiata, diploblastica, bilateral and triploblastica, right? So let's begin with our meaning, right? So let's begin with the body symmetry. When I talk about the body symmetry, class symmetry you must have studied in the lower classes. In maths subject usually we study. Suppose if I'm going to draw a butterfly over here and I'll ask you, is it symmetrical? So I am going to draw a plane from which is going to pass through the center, and I'll check one is similar to 2. Yes, one is similar to 2, so we call it as symmetry. So symmetry is nothing but when any plane pass through the center, it divides body into two equal halves, right? So symmetry is nothing but when any plane or a plane passing through, a center, it's going to divide body into two equal halves. We call it as symmetry. So based on that, we have three types of symmetry: asymmetric, radial symmetry, and bilateral symmetry. When we talk about asymmetry, A means what? That means absent. Asymmetry means there won't be any symmetry. So no matter what number of plane you are passing through the center, body won't be able to divide into two equal half. For example, let me draw a rough diagram over here. Suppose this organism I have taken, maybe this is porifera. So no matter I'm going to pass through this or this way, or any plane I'm going to pass through the center, it is not going to divide body into two equal half, right? Now let's jump on the second one, that is radial symmetry. Let me draw it over here, it will be better. Okay, now when we talk about radial symmetry is nothing but, suppose this is an organism and I am going to pass, better than this, I think I can go with, okay, it's not there, let it be. Okay, so if I am going to pass plane from center, this is my center, one plane, let me pass another plane from the center, let me pass two more planes, another one from here and another one from here. No matter how many plane I will be passing, class, body will be able to divide. Okay, I'm sorry, no matter how many planes I will be passing from the center, in asymmetric classification, body won't be divided into two equal half. But when it comes to the radial symmetry, any plane passing through the center, is going to divide body into two equal half. Whether you take this plane or purple one or green one or blue one, whatever plane you are going to take, and it should pass from the center. Criteria is it should pass from the center. The body is divided into two equal half. Then the last one is bilateral symmetry. What is bilateral symmetry? Bi means what? Two. So let me draw somebody's body over here. This is my favorite student, Yagna's body. Let me draw cute eyes. Okay, fine. Now class, bilateral means, bi means two. So a plane, mind it, I said a plane, okay? A plane passing through the center, it is going to divide body into two equal half. First and second, both, if you will see, both are equal. Am I passing many plane? No, I am passing only and only one plane. So a plane passing through the center is going to divide Yagna's body into two equal half. This type of symmetry we call it as bilateral symmetry. Which kind of symmetry it is? Bilateral symmetry, right? So let's come back and see our example. So here you can see, class, radial symmetry is nothing but when any plane passing through the center is going to divide body into two equal halves. Not R. Okay. Examples are some of the sponges, Coelenterata and Ctenophora. What did I tell you in the class? Coelenterata and Ctenophora both are best buddies. Wherever Coelenterata will be there, your Ctenophora will go there, right? Adult Echinodermata is an example of radial symmetry, while larva Echinodermata is an example of bilateral symmetry. It is a neat question, class. You have to focus on this point, right? Now, bilateral symmetry means, bi means two. Whenever a plane, I have not written any plane, a plane passing through the center is going to divide body into two equal halves, right? That we call it as bilateral symmetry. Let's see few of the examples. See here, so this is an example of porifera. Here, no matter which plane you are passing through, you won't be able to cut this organism or divide this organism into two equal half, no matter from wherever you pass the plane, right? So this is an example of asymmetry. Then we have radial symmetry. Here you can see class, this is the example of starfish. I am passing plane from the center. A plane is passing from the center will be able to divide body into two equal half. But is it going to be a plane or any plane class? It is going to be any plane. Whether you pass from this way or you pass from this way, any plane passing through the center is going to divide body into two equal half, right? Then what is the example of bilateral symmetry? Here you can see the example of housefly. A plane, mind it, a plane. I'm going to pass a plane through a center and it is going to divide body into two equal half. But if I'll pass plane like this, will the body is divided into two equal half? No, the body will not divide into two equal half. That's why I said in bilateral symmetry, we are not supposed to use any plane. We have to use a plane, right? A plane passing through the center is called as bilateral symmetry, right? So that was all about the symmetry. Now let's jump on the another basic topic, that is our germ layer, right? What is germ layer class? I have already discussed with you what are germ layers. Let's come back to the flow chart and then you will better understand. So during fertilization, what happens? Sperm and egg are going to fuse together, and by the process of fertilization, what is formed? Your zygote is formed. How many cell zygote is? It is one cell, right? One cell. This zygote is further going to develop and form your morula. Morula is further going to develop and form your blastula. Blastula is further going to develop and form your gastrula. And gastrula, and the process of formation of gastrula, we call it as gastrulation, right? But before moving ahead, let me explain you a little bit more about morula and all. So class, this is your fallopian tube. This is your fallopian tube, right? Now here, fusion of sperm and egg takes place, right? So there will be the formation of zygote. Zygote is there. Zygote divide into two cell. Now, by which process it divides? By the process of cleavage. What is cleavage? Cleavage is a special type of mitosis. Why we call it as special type of mitosis? Because there won't be any G1 and G2 phase. So, cell number will increase, but the size of cell won't increase. So, this is 2 cell. Then we have 4 cell. Then we have 8 cell. Let me draw 8 cell. And then we have 16 cell, and then we have 32 cell, and then you have uterus over here. So this 8 to 16 cell, we call it as morula. Cells are tightly packed, which gives you mulberry-like appearance. Mulberry-like appearance. Okay. Now, can you see over here, have I increased the size of the cell? No, I haven't increased the size of the cell. Only the number of cells are increasing. So, we call it as morula. Now, do we have any cavity till now? No, we do not have any cavity. Cells are compactly packed. So, these are 32. Cells keep on dividing and more than 100 cells, and then it jump into the uterus. It jump into the uterus. Once it jump into the uterus, the uterus is going to produce uterine milk, which is going to enter inside the cell. So what will happen class? The cells which were present inside will start arranging themselves. In such a way that the fluid which enter inside will make its space and it makes cavity. Now this cavity is going to be fluid filled cavity, and since it is present in blastula, we call it as blastocele. Cele word we use for cavity, right? Now this cell we call it as blastula. Okay. Now this blastula is going to embed with the uterus, and further development of this blastula takes place. So what is the further development? After blastula, there will be formation of gastrula. What is gastrula? Gastrula is nothing but formation of germ layer. So now finally your germ layer will be formed. What is germ layer class? Germ layer is a layer through which your entire body is formed. Your organ system, the whole organism is formed from these germ layers. Now some of the organisms will have 2 germ layers and some of the organisms will have 3 germ layers. So basically we have 3 germ layers. We call it as endoderm, that is inner layer, ectoderm, that is outer layer, and then we have mesoderm, that is middle layer. This process of formation of germ layer, we call it as gastrulation. So what is gastrulation? It is nothing but process of formation of germ layer. We call it as gastrulation. So I hope there won't be any confusion between blastula, gastrula and morula. So in morula there is no cavity, blastula cavity will be present, and gastrula you have germ layers. Okay, germ layers are not present in blastula. What are germ layers, class? Germ layers are nothing but these cells are going to arrange themselves in such a way that it will become germ layer, and these germ layers are going to form your body. Right? Now, let's come back. So, see here class, through the process of gastrulation, and these are the three germ layers. But let's see what we have written. See here class, at the stage of gastrulation, what is gastrulation? It is nothing but process of formation of germ layer. Here in the picture you can see class, this outer one we call it as ectoderm, middle one we call it as mesoderm, and inner one we call it as endoderm. Can you see how beautifully inner one is invaginating inside, and this is nothing but the movement of the cell, rearrangement of the cell occur inside for the formation of the organs, right? Now cell undergo group movement that leads to the repositioning of the cell. This repositioning of the cell is very important in order to form the whole organism, and these repositioning of cell will interact with each other, right? The resulting cell layers that form is called as embryonic germ layer. So nothing but blastula cells are there. These cells are going to arrange themselves, reposition themselves and interact with each other, and now they are going to be called as germ layer, right? Germ layer are the one who is going to form the body of the organism, right? So let's come here. Endoderm, if we'll talk about, endoderm is the inner layer. It is going to form the inner lining of alimentary canal. For example, suppose let me draw it over here, mouth, your stomach, and then we have intestine over here, right? So all these inner lining will be made by your endoderm. Inner lining will be made by endoderm. Then what about mesoderm? Mesoderm is the middle layer. It is present in between alimentary canal and body wall, right? Maximum part of the body is made up of mesoderm, which forms organ. Ectoderm is nothing but your outer layer, and it is going to form outer part of the body. For example, your skin. For example, nervous system. Right? Nervous system. Mesoderm example, if I want to take, I can write your muscle. M for mesoderm, M for muscle. Then you have blood vessel. Let's see an example of germ layer over here. So I have taken an example of intestine. So this you can see it is an intestine, and I have taken the cross section of intestine. See here I have cut it over here. You can see this outer layer blue color, and then inner half this you can see blue color, and inner one, can you see the innermost one is your yellow color. From here only your food is going to pass. Okay, this is your inner layer. This is the lining of endoderm. So over here if you see the picture in enlarged form class, let me enlarge it more if I can. Okay. So here, this yellow color one, see. Can you see the hole? Through it, your food is going to pass. This is the inner side of your intestine, right? So it is covered by this yellow color which you can see here. That is nothing but your endoderm, right? Outer covering we call it as ectoderm. This blue color one which you can see here, this is your ectoderm, right? Now, this, let me make it smaller. Okay. Now this third one, the cavity which you can see here class, beautifully you can see here, here is the cavity. This cavity, can you see this purple color one or maroon, whatever color you say, this is nothing but your mesoderm. So this cavity all over you can see that this cavity is lined by mesodermal cell, right? This cavity is lined by mesodermal cell, so we call it as coelom. I hope here onwards you will understand. See here beautifully the cross section of small intestine, and I have explained it over here, right? Now, based on these, we have two types of organisms. What are the two types of organisms? They are nothing but your animals are classified based on embryonic layer into two types of organisms. One is your diploblastic and other one is your triploblastic. When I say diploblastic, di means two, tri means three, right? So these are the organisms who have only two layers. What are the two layers? Outer and inner one. Class, this is, suppose this is your endoderm, right? And this is your ectoderm. Cell from endoderm will come out and it will form another layer. We call it as mesoderm, right? So it is not compulsory that mesoderm will be present in each and every cell, correct? Endoderm and ectoderm will be present in each and every organisms, right? Now, external, ectoderm, internal, endoderm. Then what is present in between endoderm and ectoderm? That is mesogelia. What is mesogelia? Is mesogelia is a cell? No, it is not a cell. It is non-cellular or acellular gelatinous fluid. Is it differentiated or undifferentiated? It is undifferentiated. What is the meaning of undifferentiated? It does not perform any function. Okay, let me write over here. Does not perform any function. If a function is assigned to a cell, we call it as differentiated layer. For example, your porifera and your best buddy coelenterata and ctenophora will come under diploblastic. What about diploblastic? All the three germ layers will be present. Example from platyhelminthes to chordata, right? Now see here the examples class. Here you can see the diagram. This purple color one is nothing but your ectoderm and this is nothing but your endoderm and this is your gut. This is what I told you. For example, that alimentary canal is there. Na? For example, this is here, right? So this part, this empty one which you can see, I have written gut over here. Okay, I have written gut over here. So this is your gut, endoderm, and this is your endoderm. What about this red color one? These are mesogelia, like gelatinous substance. Let's see about triploblastic. Ectoderm is present, this purple one. Then we have another, this endoderm, and in between ectoderm and endoderm, we have mesoderm. Okay, so this was about your germ layer. Here you can see how the beautifully they have arranged. This blue color one, you can see this is your ectoderm and endoderm is going to invaginate inside in such a way that the cavity is formed. This cavity has name, we call it as archenteron. It is nothing but your hollow cavity. Archenteron is nothing but your hollow cavity. What is archenteron? Archenteron is a hollow cavity lined by endodermal cell. Which cell? It is lined by endodermal cell, right? Now, suppose this is your ectoderm and this is going to be your endoderm, right? Now, this opening, this pore which is present over here, we call it as blastopore. The cavity which is formed inside, this is the cavity, yellow color one which you can see here, this is the cavity which is formed inside. This cavity we call it as blastocele because I have taken example of blastula. What did I tell you? Gastrula is nothing but the cells which are present in the blastula, they are going to rearrange themselves, right? So rearrangement will occur and then you have formation of cavity inside, right? I hope I am clear. So that was about your germ layer. Let's see about the archenteron. Archenteron is nothing but a hollow cavity inside the gastrula which is lined by endoderm, right? And it has blastopore. Blastopore is the pore is what class? Pore means hole. Blastopore is the one who has capacity to form anus or mouth. Okay, it can form either anus or mouth. Created by invagination. What happened? This endoderm will invaginate, move inside. This endoderm will invaginate or move inside to form archenteron. Function develops into digestive tract. This only, this is gut na class. So this is going to develop into your digestive tract. Then what is blastopore? Blastopore is nothing but opening of archenteron. This hollow cavity which is present inside, this hollow cavity I call as archenteron, right? Archenteron. So this opening of archenteron, this part I call it as blastopore, right? Which will eventually become either the mouth or anus, correct? So that's all about your germ layer. Now let's jump on the another topic, that is phylum, a beautiful cockroaches dancing, like students are dancing in my class. Okay, I think I should keep name of this cockroach, but let me ask what name I can keep. Anyways, jokes apart, let's study about the phylum. These 11 phylum name you are supposed to mug up class. So we'll start with the phylum Porifera. First one is Porifera, then we have Coelenterata or Cnidaria, then we have Ctenophora. Now, usually we get confused, Coelenterata or Cnidaria, or it is Ctenophora or Cnidaria. So always remember class, my Ctenophora, Tina is very, very special. It doesn't want any other name. Okay, so it's special, specifically exclusive exclusive. So there won't be any other name for ctenophora. Now phylum Platyhelminthes, then Aschelminthes, phylum Annelida, phylum Arthropoda, phylum Mollusca, phylum Echinodermata, phylum Hemichordata, and phylum Chordata. These all phylums line by sequence wise you are supposed to remember it, right? Now, acha. Also, how you can remember it, class? I remember it. P for porifera. Then we have C coelenterata. Now, C coelenterata, O and N will come before then T, right? So, coelenterata, C square. C, one C is coelenterata, another one is your ctenophora. Then we have platyhelminthes, and then we have ask. Ask to whom? A cube. Ask to whom? Ask to Anil. Any, you can say Annie or Arthro. Ask to Anil or Annie or Arthro. N will come before and R will come later. Ask Annie, N will come before, R will come later on, and Mehek. Mehek, okay. PC square, PA cube, after square cube will come, like you can remember Papa will be there na, or PC Pa, PC square Pa cube, PC square Pa cube, Mehek is the trick for this thing, right? Now, let's come back to our topic, that is classification of kingdom Animalia. When we talk about the kingdom Animalia is divided into two subkingdom, Parazoa and Eumetazoa. We have already seen all these things. Let's differentiate between Parazoa and Eumetazoa. Parazoa, para means primitive and zoa means animal. Eu means true and zoa means animal. Are they both multicellular? Both are multicellular. Is there formation of any tissue? There won't be any formation of tissue. What about this? Eumetazoa, there will be the formation of tissue over here. No higher level of organization. अगर tissue ही नहीं है, if cells are not going to aggregate to form tissue, so obviously you are not going to see organ or organ system. But here tissue is going to form. So this tissue is going to combine together to form your organ and organ system. So you will see high level of organization over here, but you won't see in Parazoa. Right? Example is your porifera, sponges, and except porifera, all the animal kingdom will come under Eumetazoa. Subkingdom Metazoa is included, it's going to divide it into two grades. What are the two grades? Radiata and Diploblastica, Bilateral and Triploblastica. Now, since we have already seen about the types of animals and symmetry, can anybody guess radiata means what? It's going to be a radial symmetry. Bilateral means bilateral symmetry. Diploblastica means diploblastic animal. Triploblastica means triploblastic animals, right? Let's see over here. So when we talk about radiata or diploblastica, it is nothing but they are the first true metazoan or eumetazoan, and they have radial symmetry. I already told you, any plane passing through the center is going to divide body into two equal parts, right? Then diploblastic, only and only two germ layers are present. What are the two germ layers? Your ecto and endo. There is no mesoderm present over here, right? Example is your Nidaria and Ctenophora, your best buddies, right? Here you can see example class. Aurelia, jellyfish, how beautifully it is moving, and your comb jelly. Next one is your bilateral triploblastica. Bilateral triploblastica is nothing but again, triploblastica means three germ layers are going to be present, and bilateral means a plane passing through the center is going to divide body into two equal parts. That's what we have already studied. Now, bilateral includes two divisions. What are the two divisions? These are protostomia and deuterostomia, right? Now, let's understand about protostomia and deuterostomia. Now, we are going to differentiate between them. Proto means what? Proto means first. Stomia means mouth. Deutero means second or secondary. Means लिख दिया है, means है, okay, MEANS, means secondary. Stomia means mouth, right? So here blastopore, I hope you remember, blastopore is nothing but the opening of archenteron, right? Opening of archenteron, and I told you that blastopore has the capacity to form anus or mouth. So when we talk about proto means first, stomia means mouth. So can anybody guess what will happen over here? Mouth is going to form first, and who is going to form? Who is going to help? Blastopore is the one. So your mouth is going to form through blastopore. Mouth develops from blastopore. So these blastopore is ultimately converted into mouth, and these endoderm is going to invaginate more, and here it comes and forms your anus. It is going to divide. Again, pore will be created. Okay. When endoderm is completely going to grow, it is going to create another pore which is called as anus, right? Anus is formed later on, that is away from the blastopore. What about deuterostomes? Deuterostomia means blastopore is going to develop into anus. Deutero means second, stomia means mouth, that means mouth is going to form second, right? So here anus developed from blastopore. Now, what type of cleavage will be seen here, class? It is going to be spiral cleavage. Just see here, one zygote and then divided into two cell divided into four cell. Here, can you see 1, 2, 3, 4? All cells are not aligned with each other. Then 5, 6, 7, 8, they are not one above the other cell. They are in the twisted form, in the form of spiral. You can see here, cells are not placed one above the other. But on the opposite, if you see, zygote is divided into two cell, four cell, and here can you see this cell? This cell number 1 is above the cell number 2. Cell number 4 is above the cell number 3. So cells are one above the other. They are not in the form of twisted. This type of cleavage we call it as radial cleavage. Now, how is the cell development in protostomia? It is going to be determinate. What is the meaning of determinate? Now, let me show you the picture and explain. See here. So, these are the cells present in the protostomia. Now, if I am going to excise one of the cell, my embryo is not going to form properly. So, development will arrest. Why this happens? Why this happens? Because based on cell position and molecules, function of each cell is predetermined. That means each and every cell over here will have its own function. That cannot be, suppose one is performing some function that cannot be performed by cell number 2. Cell number 2 function cannot be performed by cell number 3. So they have their own predetermined function which cannot be performed by other cell. So if any earlier cell is removed or excised, other cell can't take over its role. Due to that, what will happen? Complete organism won't be formed. And this type of formation of embryo, we call it as mosaic embryo. Mostly where you will see this in invertebrates. Then what about indeterminate cell division? Sorry. Indeterminate cell development. It is nothing but, let me show you the picture. So again here I have cell 1, 2, 3, 4. Even if one cell is excised, what will happen? Suppose if I am going to remove cell number 3, the function of cell number 3 can be performed by cell number 2 or cell number 4 or cell number 1. Okay, that means here whatever embryo is going to form, it is due to cell interaction. Okay. Let's see over here. Early embryo can regulate themselves even after the cell loss. Cell have more flexibility. Their final fate, that means finally what embryo is formed, is determined by cell to cell interaction. Now let's see. If in early cell, if I am going to remove any of the early cell, other cell compensate and take on its developmental role. So one cell, if I am going to remove, the other cell will be present who is going to support, who is going to play the function of that removed cell, right? Hence this type of embryo is going to be normally, and this we call it as regulating embryo. This type of embryo we call it as regulating embryo. Where will you see this type of cell development? You will see mostly in vertebrates, right? So here you can see schizocoelomate. Protostomia are schizocoelomate and deuterostomia is enterocoelomate. Just see this diagram. I will be explaining you schizocoelomate and enterocoelomate little later on. See here what happened. Cleavage is there. From cleavage blastula is formed, and here you can see, class, how endoderm, you have to draw this diagram, class, how endoderm is invaginating, and from endoderm, that is from the lip of the blastopore, a cell is coming out and it forms mesoderm, right? Here you can see whole endoderm has invaginated, and due to this what happened? Division, or the pore will be created over here, right? Pore will be created over here. The second pore we call it as anus, and first pore was blastopore, from which mouth is formed, right? Let's see for the second one, deuterostome. When we see deuterostome, what happened? Endosperm is going to invaginate, and from endosperm there will be the outpocketing or pouch formation will be there. This outpocketing or pouch is called as mesoderm. Okay, this is called as mesoderm. And then again here we have blastopore. This blastopore is going to convert into anus. This is going to convert into anus, and second mouth is going to form. Now in chat box, quickly write and tell me, are we protostome or deuterostome? What is formed first in our body, anus or mouth? Now here clearly you can see, class, spiral image is there, and beautifully endoderm is going to invaginate and a cell is coming out of it and mesoderm is formed, right? Fine. You have already seen this. We have already discussed this. I thought of explaining cystocoelom and enterocoelom, but let it be. Okay. Now, protostomia division protostomia includes three subdivisions. What are the three subdivisions? Acoelomata, pseudocoelomata, and schizocoelomata. So before explaining all these things, I should explain you what is coelom. Let's jump over here. So here is a picture of your alimentary canal. So this, it starts with mouth and ends with your intestine or rectum, or I can say anus, right? So that is your alimentary canal. The other name of alimentary canal is your gut. Now, when you see, this is your body, and this which you can touch, which you can see, we call it as body wall. And here inside you have your alimentary canal, right? Now see here class, here you can see your alimentary canal, this inner body part you can see alimentary canal, and here is the body wall. Now in between body wall and alimentary canal, can you see the cavity? This cavity is filled with fluid. This cavity is filled with fluid. This we call it as body cavity or coelom, right? So class, here we can see 3D image of our alimentary canal. That is, it starts with the mouth and it ends with the anus. From back side you can see mouth, esophagus, and esophagus is going to enter into your stomach. Stomach is going to enter into your intestine, right? Now here you can see class, this hole is your alimentary canal and this is your body wall. Can you see there is a cavity? Suppose if I talk about this intestine, I hope you can see my cursor. So can you see that there is a gap between body wall and intestine? Actually this gap is nothing but there will be the fluid filled cavity. This fluid filled cavity, if it is lined both the side with the mesoderm, we call it as coelom, right? So fluid filled cavity, when it is lined by both, at both the sides, at all over the side, if it is lined by mesoderm, we call it as coelom, right? So here all over you can see we have body cavity present in our body, right? Class, this is called as body cavity. Here is your alimentary canal. See your stomach, your intestine, everything is there, and this we call it as, all these things are your body cavity which is present inside. In between the organs, body cavity is present which is filled with fluid. So what is body cavity? Body cavity is nothing but which is going to fill with the fluid and it is lined by mesoderm. So what is coelom class? What is coelom? Coelom is nothing but it is a body cavity. Which cavity? Fluid filled body cavity which is lined by mesoderm. Fluid filled body cavity which is lined by mesoderm, right? We call it as coelom. Now see here class, let's see the picture and you will understand in a better way. This blue color is nothing but your ectoderm. This yellow one is your endoderm. Again ectoderm, endoderm, ectoderm, endoderm. This much is clear to you, right? Now let's see about acoelomate. Coelom, what did I tell you? What did I tell you? That is, coelom is nothing but body cavity filled with the fluid. When I say A means there is no body cavity present. You can see in between ectoderm and endoderm you have cells. This pink color is nothing but your parenchyma cell or mesenchyma cell. What is mesenchyma cell? These are nothing but derived from your mesoderm only. Cells which are derived from your mesoderm. Let's come to our next, that is pseudocoelomate. Coelom I already told you. Pseudo means what class? False. Let's see over here. So again you have ectoderm and you have endoderm. This is going to be your gut. Okay, you have ectoderm and you have endoderm. When we see ectoderm and endoderm, this pink color one is your mesoderm. Okay, mesoderm. Now class, at one side mesoderm is present, but do we have mesoderm at other side? What did I tell you? For a coelom, suppose if this is called to be called as coelom, everywhere it is supposed to be surrounded with body cavity. Okay. So now this, sorry, my bad. I said if this is supposed to be coelom, then body cavity is supposed to be surrounded all over through mesoderm, right? So here at one side mesoderm is surrounding, but what about the other side? Mesoderm is absent. So we felt like that ki, okay, it is a coelom, but later on we understood ki no, it is not. The body cavity is not lined both the side from mesoderm. That's why we call it as pseudocoelom. What about coelomate? Coelomate is nothing but, see here, ectoderm, endoderm. Here you can see class, this is your body cavity. Body cavity, or we call it as coelom. Body cavity, or we call it as coelom. Can you see class? Here also you have this pink color of mesoderm, pink color of mesoderm, pink color of mesoderm, pink color of mesoderm. Everywhere it is lined by mesoderm. So to organism to be called as coelomate, body cavity has to be lined all over by your mesoderm. Am I clear? Now, let's see what is written in your textbook. A means without, coelom means body cavity, and atta means having. So, these are first triploblastic eumetazoan and they have solid body plan. That means there is no cavity present inside. Instead of cavity, what they have? They have parenchyma cell or mesenchyma cell. Mesenchyma cell derived from mesoderm. Now, what is perivisceral space? What is perivisceral space? So class, we already discussed viscera means what? Internal hollow organ. Internal hollow organ. So space in between the viscera, space in between the organ, we call it as perivisceral space, right? Example is phylum Platyhelminthes, that is your flatworm. Let's jump on pseudocoelomate. Pseudo means false, coelom means body cavity, atta means having. So animals of this group possess body cavity between body wall. Now, let me move back here. See here. So body cavity is present. This is your body wall and this is suppose your stomach. So can you see this body cavity over here? So body cavity is present over here. But when we talk about pseudocoelomate, it is not lined both the side. It is not a true coelom. Why? It is not lined by mesodermal epithelial layer. That means parietal and visceral layer. Parietal class, one example if I want to give you. This is suppose your, let me draw it again. This is your lungs. Okay, so lungs have two layer of covering. Now when we talk about two layer of covering, so what did I say? Internal organ is viscera, so lungs is a viscera. So internal layer we call it as visceral layer and outer layer we call it as parietal layer. Outer layer we call it as parietal layer, right? Example is your nematode. Now, sizocoelom means what class? Whatever we saw for protostomia, same thing sizocoelom. It's just that here, what did I tell you? From the lip of blastopore, cell is going to come out and it is going to form mesoderm. Can you see when the mesoderm is splitting? Earlier, what will happen? Mesoderm is going to be like this, no splitting. But later on, it splits. And when it splits, there is a formation of cavity. So you can see cavity over here. Okay, we call it as sizocele. Sizocele. Okay, the cavity is called as sizocele. So sizocele is formed by splitting of mesoderm, that is your outer somatic cell. So this layer, the outer layer, we call it as somatic cell. Somatic layer, and inner layer we call it as splanchnic layer. Okay, so inner layer we call it as splanchnic layer and outer layer we call it as somatic layer, right? The example is your phylum Annelida, Arthropoda and Mollusca. Now, your deuterostomia, it includes one subdivision, that is your enterocoelomate. We have already seen enterocoelomate. They are true coeloms. That means they have enterocele. These are the examples. In deuterostome you will see this class. So here, see here class. Now this, why do we call it as enterocoelom? What did I tell you? They are going to form, suppose this is your ectoderm and this is your endoderm. Endoderm is going to form lining of your alimentary canal. In alimentary canal, we have intestine, right? Intestinal cell, we call it as enteric cell. Intestinal cell, we call it as enteric cell. So these enteric cells are going to be out pocket. Okay, so this out pocket which is coming, it is the enteric cell. From that we have given name entero. Okay, so the cavity which is present over here, we call it as enterocele. Example, your phylum Echinodermata, phylum Hemichordata, phylum Chordata, right? Here you can see example class, same thing what we studied in protostomia and deuterostomia. Same thing we did it. Lip of the pore, your cell will come out and it is going to form mesoderm. Mesoderm will split and it will develop coelom. This coelom we call it as sizocele. This coelom we call it as enterocele, right? So that's all we have in today's class. Thank you.